Okay, so I was told to start the celebration with something that would get your attention. So I hope everybody hears that. And we have over here a table that looks like it's set up for cake, but we need a cake. And so here's the cake. Now, Francis's role is only to get the cake close to the table, but then he's going to join me over here. When it gets dropped on the floor, it wasn't me. No. So thank you to Dave Kaufman for picking up the cake this morning and safely getting it here in one piece.
You know, we had a plan. There was going to be a backup cake, and I was going to come in and drop it on the floor, and then we were all going to look at your face, but we didn't quite pull off the backup.
And that would have been funny.
Yeah.
Not. So, Francis, first of all, never once did you ever have glow sticks at a council meeting? We're celebrating. Do you even know what a glow stick is?
I think so, yeah.
You have grandchildren. Oh, look. And he even knows that he can make it into a bracelet. Look at that. Okay.
Yeah, and it makes colors.
It makes colors. It will glow. There you go. That your wrist, it may not fit on your wrist.
Yeah, I'm a little bit large for
So,
this, but
okay. So we have we have cake, which is really cool. We have a special guest, which is also really cool. And let me set some context about what we're celebrating. We are celebrating 100 and counting meetings of the National Advisory Council for Human Genome Research. Now, that's why we have Francis here to help celebrate this. That's why we have cake. But I thought it might be fun to do a few other things associated with the celebration. First thing is to remind everybody sort of the history of the Institute with respect to who has sat in this seat and presided over these council meetings as the NHGRI director. So during, and we'll do a count along the way. So during, as you heard during my director's report, Jim Watson was not available to preside during the very first meeting of this group, and longtime Deputy Director Elka Jordan stepped in, and her total number of times presiding over council as the NHGRI director was once, but she gets on the list. It was a big one. Then Jim took over the Institute, and Jim was present for 3 meetings of this group. So 1, and then 3. And then we went to interim leadership. Suspense, there's Michael Gottesman, who was the acting NCHGR director for a total of 3 council meetings and remained and is still a great friend and colleague of the Institute. Michael then went on to become, for many years, the deputy director for intramural research at the NIH. And then this guy came. There's a young and a little not as young, and then there's here.
only 46?
46 council meetings.
Felt like at least 47.
Okay, so 46 council meetings. But then, as you know, because you've all followed the history of the Institute, Francis departed, and Alan Guttmacher took over as the acting director of the Institute. And he presided over 4, so that was more than Gottesman or Watson. And then the baton was handed to me, and knowing that Francis had 4, but today is my 43rd. So
Got to work harder.
there is fierce competition between Francis and I, but I know I'm data driven. I face reality. I appreciate that Francis has done 46. I've done 43, and 46 is more than 43. So Francis remains the greatest of all time.
All right.
As we learned during the competition based earlier this year at DNA Day, where he emerged as the GOAT of the Institute, and he continues to be the GOAT at least for 3 more council meetings, at least then when I'm gonna start to tie. So we are here today with Francis here celebrating. This is an image that is on the cake. You may want to look at the image because by the time you get in line to get a piece of cake, it may not look like that anymore. Although Ernesto's going to make sure to get photographs of the cake, photographs of the decorations. No one's cutting the cake until we take some pictures, but I wanted to do that. So Francis, did you want to make any general comments about this being the 100th meeting of this advisory council?
Well, I heard there was a party, so I wanted to show up for that, and I wasn't sure whether maybe you were declaring you'd finished the human genome again, but I guess maybe we've finally come to the end of that series of accomplishments. Oh, well.
No, now we do telomere to tail. We just announced the Y chromosome.
Well,
Again.
okay, there must be something else. You can still do one more or two more of these to say, we're done, we're really done. No, I wanted to be here for the 100th. And again, I was only 46. I was guessing I had probably hit 50, but I'm sure your count is absolutely accurate.
Oh,
No,
yes, just like precisely finishing the Human Genome Project precisely when the human sequence was done. Yes. We never shift odometer moments, ever.
No, I'm really glad to see all of you. And thank you, council members who have not been here for 46 or 43, but probably for enough that you've done a lot of work, including at this particular session. And that's the way that this whole amazing Genome Project and its institute have gotten done, is by that incredibly capable advice. And thank you, staff. It's great to see all of you sitting there smiling, and many of you who've been laboring through many of these council sessions and making it all come together. I'm really proud with the way in which this part of NIH continues to be right out there on the leading edge of all the exciting things that are emerging now. Everything from basic science to clinical applications and social applications. So it's good to see you all in this different space that I hadn't even been to before, even though I guess you've been here for years.
I had to tell him where to park. I sent him photos this morning. I was so worried he wouldn't know where to come.
He sent me 3 pictures of the street corner and the sign and the picture of the entrance to the parking deck, because he obviously thinks I'm clueless about how to find my way around.
No, but when you were NIH director, you were high maintenance and you had to really take good care of you. I should have realized that, you work for me now in the intramural program. I probably don't need to take such good care of you.
Yeah, well, I'm glad you still had the right instinct.
Francis, I just thought of something. I had not thought of this before, but I vividly remember you telling me when you handed me the baton and I accepted the job after you offered it to me. I remember among like the 2 or 3 key things you told me. I'm being serious here. I remember you telling me that one of the most gratifying parts of being NHGRI director will be interacting with the advisory council. And I didn't really understand what that meant. I sort of could imagine because I had worked with my intramural board of scientific counselors. And so I sort of got a sense. But I remember you said those would be some of the most stimulating, incredibly gratifying interactions. And, absolutely 40 plus now, meetings into this, I completely agree with you. And it's this group, but it's all the people that have sat in the seats before, including the ones that were in place even when I got there that I did, or I wasn't the one appointing them, but it's been incredible. These 3 meetings a year are some of the most stimulating days of the year. You want to make it, because you felt the same way during your time as director.
I totally did, and 27 institutes and centers, they don't all operate the same way in terms of how the advisory councils function, and some of you who may have even been on other councils will agree with that. I see some nodding heads. From the very beginning, the Genome Center and then the Institute saw this as just a golden opportunity to bring people with big ideas, big vision, big willingness to take on hard problems and try to steer the effort and really put them to work. And that has worked beautifully for a circumstance where, genomics is changing overnight. So you really got to have that kind of wise, flexible, on top of things advice. Not a lot of the other institutes, I think, have had quite that same attitude. Frankly, I think some of the institutes, councils are sort of rubber stamps. You have a nice presentation on something, and then you have some sort of en bloc, and you go home. This council has been certainly asked to be much more engaged from the very beginning, and it has served science really well.
And one last thing, because I get asked this all the time, but you have a chance with all the council members and people watching virtually and so forth. So what are you doing lately? Idle time is the devil's playground. Are you keeping busy? Are you getting in trouble? I don't supervise you, nor do I supervise the person that supervises you. You're like 2 or 3 levels down at the Institute.
And that's okay, by the way.
I just want to get
really
a sense.
fine.
I mean, just share with everybody what you're doing. I know your lab is being site visited next month. I know you're working hard on that. Are you goofing
quite a bit.
otherwise?
No, I am happy to be a intramural principal investigator in the Center for Precision Health Research, which is a new part of the intramural program with Les Biesecker as the chief. So I report to Les, which is interesting because he used to report to me. But, these things get turned upside down all the time. And then he reports to Charles Rotimi, and I guess eventually this gets to you, but only if we screw up really badly. Otherwise, you're not supposed to hear about it. My lab is about 12 people. We are working on diabetes, epigenomics, doing a lot of really cutting-edge single-cell RNA-seq and ATAC-seq with stem cells that we differentiate into insulin-secreting beta cells, and a lot of CRISPR stuff. And my lab is also engaged in developing a clinical protocol for in vivo gene editing for progeria, this rarest of all causes of premature aging, which we hope in the next year or two to actually have that into patients. And this will be pretty bold, a single intravenous infusion of a gene editor engineered by David Liu that we think should provide significant benefit by fixing that one nucleotide that's misspelled in Hutchinson-Gilford progeria syndrome. So we have a great time in the lab. Larry Brody is in the same lab space as I am, so his group and my group bump elbows a lot. And it's pretty exciting in Building 50 right now, all the things that are happening with precision health. On top of that, I did spend 15 months in the White House as the president's acting science, but yeah, right. Hard to find your way out of the West Wing. And I'm still charged by the White House, including meeting with the president on Thursday, to lead this effort to eliminate hepatitis C in the United States. And I am obsessed about this, and you're going to be sorry you asked, but I'll try to keep it short. Basically, this is a disease that kills 15,000 people every year, for which we have a cure. A really simple, easy cure. 1 pill a day, 12 weeks, 98% cure rate, almost no side effects. And yet 2.5 million people in the United States who are infected with hepatitis C are not getting access to that cure because of our broken healthcare system and the cost of the drug, and that is just not right. So it should be possible, if we had a political system that was sensible, which is something we'll have to talk about, to convince even a skeptic that you want to do this to save lives and save tens of billions of dollars that won't be needed for all those liver transplants and all those liver cancers that will now not have to be treated because they won't happen. This is the most common cause of liver cancer and one of the most common causes of needing a transplant. And we estimate $44 billion will get spent just by the federal government in the next 20 years if we don't find and cure these 2.5 million people. So I'm obsessed about that, and it's going reasonably well. And then I'm working with Charles Rotimi on a project to try to build on top of H3 Africa, something you all must know a lot about because Genome had a huge amount to do with creating this remarkable 10-year program in Africa with the Common Fund providing support. And it's time to take that to the next level. And so there is a business plan that's very well worked out for a Centers of Excellence program in genomics across the whole continent of Africa. We had an all-day meeting in New York on Friday as part of the UN General Assembly Science Summit, and it went pretty well. And I think with some funds, we hope coming forward soon from places like the African Development Bank, this might actually get off the ground as soon as next year. So I'm fired up about that. And yeah, and then I'm writing a book about truth, but that's a whole other story.
So as I knew you would slow down and become a slacker. So that's wonderful. So thank you, Francis, for sharing that. So I want to be opportunistic here. I want to take advantage of having a celebration of the 100th meeting of this advisory council, have Francis appropriately joining me for this. Betty Graham, I'm going to ask you to please come forward and sit in the seat that normally is occupied by Rudy Pizzotti.